how macrophages or neutrophils kill the pathogens. Of course, you know these basics. That was good. Okay, immunity to microbes. I was telling that this lecture is about basics of immune response against different types of microbes, microorganisms, including viruses, fungi, bacteria, and parasites. It's not about immunology of infectious diseases. Immunology of infectious diseases is something different. For example, if we want to talk about immunology of HIV, it needs a complete lecture, a 2-hour lecture. Or if we want to talk about immune response to mycobacterium tuberculosis or COVID-19 or influenza, any infectious disease needs at least a 2-hour lecture. So this lecture is just basics of immune response, different parts of immune response that you know them, how they respond to different microorganisms as a category. Viruses, bacteria, fungi, and parasites. So, first of all, we'll talk about the factors that determine the shape of immune response against pathogen. What factors influence the type of immune response against the pathogen? Then, I will talk about immune response against intracellular pathogen, and then immune response against extracellular pathogen, the main 2 categories of the pathogens, intracellular and extracellular. Haji, have a look here, please. Haji is right? Great. And how pathogens, finally, I will talk about, again, like cancer, how pathogens escape from immune response. First of all, as a reminder, we have 2 main arms in immune system, or 2 layers of immune system: innate immunity and adaptive immunity. That barriers is here indeed. Barriers, sometimes it is said that we have 3 layers. The first layer is barrier, the second layer is innate immunity, and the third layer is adaptive. But sometimes it is said that barriers is a part of innate immunity. Okay? And you saw that in barriers we have different components of immune system. And adaptive immunity, again, we have cell-mediated immunity. You know this: cellular immune response and humoral immune response. That is mainly made by antibody. But cellular mediated immune response is mediated by what? T cells. Don't we have T cell here? So mainly by cellular immune response, which is mainly TLR1, cytotoxic T cells, not only T cells. We have T cells here as well. For example, TLR1, TLR17, and TLR2. Their cytokines determine the class of antibody here. Without help from T cell, we won't have class switching, we won't have memory, we won't have affinity maturation. And in innate immunity as well, we have barriers and cellular and humoral components. Can you tell me an example of the humoral component of innate immunity? 2 examples. 2. Complement system, one another. Complement system is one of the humoral component of innate immunity, another one. We talked about in last lecture, in the previous lecture. PRR. Normal flora. Maybe. Yes, maybe. Normal flora is a cell. When we talk about humoral, we mean proteins and component of the body fluid or blood. Normal flora is cells. What bacteria? Cytokines. What else? TLR. PRR, TLR. MHC is on the surface of cell. Ya Allah. MHC is on the surface. Antimicrobial peptides. Antimicrobial peptides. In the last session, Ya Allah. Antimicrobial peptides. They are produced by neutrophils and epithelial cells, and they are proteins in the body fluids. And if they are produced by neutrophils, they are released to the fluids to kill the microorganisms. So, and again, the component of innate immunity, you know all of them. Phagocytic cells, including neutrophil, macrophage, and dendritic cells. We have granulocytes, including neutrophil, mast cell, and eosinophil. You know all of them and their functions. And NK cells and complement system, they are the main components of innate immunity, plus epithelial cells. You saw that epithelial cells have different kind of PRRs. They are the first cells of the body that's encountered, that are exposed to pathogen, any type of pathogen in the barriers. And the component of adaptive immunity, B cells and T lymphocytes. You know that we have several types of T lymphocytes, including CD8 positive and CD4 positive T cells. And for CD4 positive T cells, we have T helpers, different types of T helpers, T helper 1, 2, T helper 17 mainly. And we have regulatory T cells, yeah, as a CD4 positive T cell. And CD8 T cells, and we know that the innate immune response is the first layer. That means they are the first line of immune response that induce a response that starts to fight to protect the body from microorganisms. From the entrance, the beginning of the entrance to few hours, they can induce a protective immune response. If innate immunity cannot make a successful protection, then innate immune system will send signals to activate adaptive immunity. So that's the innate immunity that activates adaptive immunity. Can you give me some examples that innate components can activate adaptive immunity? The question is this: what components of innate immunity are involved in the activation of adaptive immunity? APCs, dendritic cells, by presenting antigens. What's the matter? So look, the first one is dendritic cells and APCs to present antigen. You can see it here. Another one, complement. How complement can activate adaptive immunity? That's right. But how? By B cells. One function of complement was providing second signal for B cell activation by binding of complement receptor on the B cell to the complements on the microbes. Okay? So they are the examples of innate immune components that are involved in the activation of adaptive immunity. So, not only innate immunity activates adaptive immunity, it determines the nature of the adaptive immune response. I'm repeating it again. Innate immunity not only activates adaptive immunity, it determines the nature of the response. What does the nature of the response of adaptive immunity? What does that mean? The nature of the response is determined by innate immunity. What does this mean? Please think about it. First, think. All of you have time to think. There is two key terms, keywords here. First one is nature of the adaptive immunity. What does that mean? And the second one, innate immunity determines the nature. There is two important concepts here. Think about it. Think about it, please. Think. Wait. Nothing, ladies. Banat. Banat. طلاب بنات بنات طلاب or طلاب بنات
طلاب.
طلاب. What do you think? What does it mean? Nature of adaptive immunity. You please.
Like coding the doms and cons.
No, first of all, the nature. What does the nature mean?
Nature, maybe it was a bacteria or virus.
No,
It has a let's say, specific response, and the allergies and parasites and wounds has its specific.
A different immune response.
Yeah.
So the nature of, thank you. I'll get back to you again after my explanation. So the nature of immune response means based on the type of the antigen, we will have a specific immune response. But as a whole, we can say we have two types of responses. Humoral for extracellular mainly, and cellular for intracellular mainly. So it means the nature, but we can go in more detail. For example, for parasites, we need a specific type of immune response, including mast cells, eosinophils, T helper 2. For virus infection, we need another type of immune response. For extracellular bacteria, we need another immune response. That means the nature of immune response. The nature of immunity, it depends on the nature of antigen. So based on the type of antigen, logically, we need a specific type of immune response. Okay? Is that clear, the nature of immune response or not? Are you asleep or are you awake? Asleep.
Sleep. It's clear.
It's clear. So, the second concept. The first concept was the nature of the response of adaptive immunity. The second one was that the nature of this response is determined by innate immunity. How innate immunity determines the nature? What does that mean? How innate immunity determines the nature of responses?
It depends on antigen. Depends on antigen that present.
Yes, we know that based on the type of antigen, we will have different types of adaptive immunity. These different types of adaptive immunity is determined by the innate immune cells. The question is this: how those cells determine the type of adaptive immunity? But why mechanisms?
By
Yes, they recognize the antigen, I know. But after recognition, they have to do something in order to determine the type of the immune response. Cytokine production. So based on the type of microbes, innate immune cells will produce different types of cytokine. And based on those cytokines, we will have different T helpers or humoral immune responses. Did you remember or not? When we say nature of immune response, indeed we are talking about the type of immune response. What type of immune response we will have? No question, no comments? So, we know that almost all of the microorganisms enter the body from barriers of the body, including skin and mucosal surfaces. For example, I'm sorry. They can enter the body from respiratory tract, both upper respiratory tract and lower respiratory tract. They can enter the body from all parts of gastrointestinal tract. They can enter the body from urogenital tract or skins and other tissues. And all of them are epithelial barriers. So the first cells that are encountered with all the pathogens, all the microorganisms are epithelial cells. It doesn't matter that it's very important to know this. That it doesn't matter what is the type of the microorganism. I mean, it doesn't matter that it is extracellular or intracellular. The main site of entrance is barrier. For example, COVID virus is a virus. It's an intracellular virus. Okay? but its entrance is from respiratory tract. So the epithelial cells of the lung are the first cells that are exposed to COVID-19. Okay? So, based on the final destination, it's very important. We say that it is said that, or I told you that, the entrance site for all microorganisms is from barriers, epithelial barriers. Epithelium, yes. But some of them stay over there, so they are mucosal. Indeed, the final location is for them is mucosal surfaces. So the mucus or the barriers are indeed the final destination. But some others pass through the epithelial barrier and go to the interstitial. Interstitial means the spaces between cells of the tissue. Okay? So extracellular, so they pass through the epithelial and go to the interstitial spaces. Some of them go inside the cells. I'll talk about them. So all of them go to the body from the epithelial barriers, and they, after entrance, they will go to different parts of the body. That I'll talk about them. So, based on the type of the site of the infection, the final destination I'm talking of the microorganism, that is mucosal surfaces, extracellular, vesicular, or cytosolic, and based on the type of the infection, we will have different components of immune response responsible to control the infection. What does that mean? Here, the type of immune response against microorganisms depends on the type of the microbe, of course, or the type of the target organ, or the localization. That immune textbooks mainly focuses on this part, the localization, extra or intracellular location. But all of these three factors influence the type of immune response against microorganisms. Not only localization, not only the type of microbe, not only the target organ. All of them together determines the type of immune response. So it's important, when we talk about immune response against any microbe, we should ask which type of microbe do you mean? And in which part of the body that microbe, in which part of the cells that microbe will go. I mean about its localization, you would ask. Is it intracellular or extracellular? You would ask the target organ of that microbe, what would be. So all these questions should you ask to talk about the type of immune response. So, have a look here again. We have both extracellular pathogens and intracellular pathogens. All of them enter the body through epithelial barriers. But some of them stay just in epithelial barriers, and some of them go to interstitial, or blood or lymphs. They are extracellular. Both of them are extracellular. The intracellular pathogens again can go to intravesicles inside the cells. There are some vesicles, some organelles inside the cells that the pathogen go inside the organelle. For example, Mycobacterium tuberculosis go inside the organelles of the macrophages. Or some others go into the cytosol. You know the difference between these types, yeah? Or not? There is an important difference between two types. Here we have intracellular microorganisms, but the microorganism is inside a vesicle. But here, it's a virus. It's not in the vesicle, it's in the cytosol. It can move in the cytosol, but most of them are intracellular pathogens. So, based on the localization, we have 4 types of, based on the localization, 4 groups of infectious agents. Some of them stay in epithelial barriers, some of them go to interstitial blood or lymph, some of them are stay in vesicular spaces of intracellular, and some of them in the cytosol. So these 2 are intracellular and these 2 are extracellular. For example, E. coli and streptococci, some of them just stay in the barriers. Or again, some E. coli can make diseases in the interstitial or blood or lymph as well. Or here we have, for example, Mycobacterium tuberculosis or lepre. Both of them are intravesicular. They stay in the vesicles of the macrophages. Or rickettsia or chlamydia, they stay in the cytosol. So they are examples of bacteria. For viruses, all the viruses are cytosolic. For bacteria, we have different types of bacteria that they are residents of different locations in the body. But for viruses, all of the viruses are in the just cytosol. Okay? And for fungi, Candida is in epithelial barrier. But Histoplasma is another type of fungi which stay inside the vesicles again. Or for parasites, we have different type of parasites again. Some helminths, worms, are staying in epithelials. Filaria is examples that go inside the interstitial and blood and lymph. Or Leishmania is intravesicular. Or Plasmodium is intracytosolic. They are examples of different microorganisms that the final destination. Those are the final destination. You know that if the virus wants to go to the cytosol, it should pass through the epithelial and blood and reach the cells. For example, HPV should pass through the epithelial and then blood and then cells, liver cells. Is that clear for you?
If the pathogen intracellular, that's mean only for the adaptive.
I'll talk about it. I haven't talked about the type of immune response yet. I'm trying to show you that we have different pathogens and different pathogens have different localization based on the intracellular spaces or extracellular spaces. We have 2 extracellular spaces and 2 intracellular spaces. But it is important to know that, for example, Histoplasma should pass through epithelial cells and then reach to the macrophages. Okay? Clear? So, if you want to talk about the type of immune response now, based on this table, we have 2 groups. This middle line, please. We have 2 groups, intracellular and extracellular. As a general, we will go into the detail in the next slides. As a general, if you want to say me which part of immune response respond to these different microorganisms as a whole here and another whole here, how would you put or which part of immune response which will respond to these different pathogens? Humor for which one? Extra. Extra, have a look here.
Yes.
So humoral is for extracellular spaces.
And cell mediated for.
And cell mediated or cellular for this part. So here is immune defense against intracellular microbes. Now I will talk about immune responses against intracellular microbes, and then I will talk about immune response against extracellular microbes. And after them, I will talk about the mechanisms that microorganisms use to evade again from immune system. There is a competition between microorganisms and immune system, like the competition that you saw between tumor and immune response. The story is same, but the difference is that the tumor is just here, and the most effective immune response to tumor is intracellular or cell-mediated, but it doesn't mean that we do not have humoral immune response. I've told you, and again, I'll talk about it. So, the main immune response, please take care, I'm saying the main immune response against intracellular immune microorganisms is cell-mediated, cellular. Okay? But does it mean that we do not have humoral immune response? Don't we have humoral immune response here?
No, we don't have.
We don't have.
Humor for extracellular.
Okay? Think about it.
When the microbes or viruses proliferated inside the cells and go out to find another host cells, so that the antibodies bind to in this stage, in this.
So, that's why I said the main immune response, the most protective, is cell-mediated. But it doesn't mean that we do not have humoral. That's why I said not only, for example, a virus, an intracellular bacteria, after killing of that cell, it will be released in extracellular spaces to infect new cells. Okay, for example, HPV, if wants to infect new cells, it kills the first target cell, and for a temporarily, it's in the extracellular spaces. And when it is in the extracellular space, so humoral immune response can fight with them. Okay, or on the other hand, the virus, for example, COVID-19, this virus COVID-19 has to pass from epithelial of respiratory tract and then go into the blood and then the target cells. Okay, for example, liver cells, kidney cells. So we have the virus both in epithelial and interstitial and blood and lymph as well. Lymphatic fluid. Okay? Get it or not? Yeah? So, first of all, I will talk about intravesicular microbes. And first of all, you know that when we talk about intravesicular microbes, they are inside the vesicles of phagocytic cells, mainly macrophages. Indeed, macrophage tries to phagocytose them, but they have adapted, they have adjusted themselves to be alive inside the macrophage. So they are called intravesicular pathogens. So we have a phagocytose microbe here, have a look here, this is a phagocytic cell, and then this is phagocytosis, and you can see that these pathogens are inside the vesicles. Okay? So we have some pathogens that are now inside the vesicles. But inside the phagocytic cells, we have some other vesicles which they are called lysosome. Lysosome is the name of an organelle. What we have inside the lysosome? Some enzymes and some bactericidal different types of enzymes. Yes, there are different types of enzymes inside the vesicles. Lysosomes, they are lysosomes. They are lysosomes. So, after ingestion of the microorganisms and after trapping the microorganisms in the vesicles, we have merging of lysosome with the, this vesicle is called phagosome. Phagosome. So we have a merge between lysosome and phagosome. in order to release the content of lysosome into the phagosome. When we have them merged, they are called phagolysosome.
Yeah, I think this is the same mechanism for the eating of the cell.
For
Eating of the cell. Eats of the cell.
yes.
When the cell needs to eat.
Yes. They are they're phagocytic cells. They are eating indeed.
Yes.
Eating and killing them. Yes, that's okay. That's correct. So this is called phagosome. This one is called lysosome. And when they are merged, they are called phagolysosome. Okay? The merge of phagosome and lysosome. So what then will happen? The first thing is that some part of this pathogen, have a look here. We are talking about immune response against intracellular vesicular microbes. First of all, I'm talking about phagolysosome formation. Phagolysosome formation means ingestion and having a phagosome and then merging of lysosome with the phagosome, which means phagolysosome. And inside the lysosome, the pH is very low. Low pH is harmful for bacteria. We are talking about immune response, the mechanisms by which lysosome is going to kill the bacteria. The story is this. For example, this is the phagosome that has ingested the bacteria. So we have a phagosome that bacteria is inside it. We have another vesicle which is called lysosome. Inside the lysosome there are enzymes, there is low pH and some other toxic materials for the microbe. So we have microbe inside the phagosome and lysosome here. These two vesicles will merge together in order to pull all the content of lysosome on the microbe. So the pH is very low, that is harmful for microbe. There are lysozyme. Lysozyme is the name of enzyme. Lysosome is organelle, but lysozyme is the enzyme. Lysozyme is indeed can hydrolyze the cell wall of the bacteria. It kills the bacteria. Besides, there are some other enzymes, including proteases, different proteases. And this enzyme again can kill the bacteria. And there is lactoferrin. Lactoferrin binds to iron and deprives the bacteria from iron, prevent the iron to reach the bacteria. Bacteria needs the iron for its biology, okay, to be survive. So the first one was phagolysosome formation. Phagolysosome formation means merging of lysosome with phagosome and pulling the content of lysosome on the phagosome. That will lead to the decrease in the pH, activation of lysosine, proteases, and lactoferrin that binds to the iron and deprives the microbe of iron. No question? It's clear? So, another mechanism, all mechanisms are called oxygen-independent killing of the microbe. We have some other mechanisms that mechanisms need oxygen. So those mechanisms are called oxygen-independent killing of the microbe. These mechanisms do not need oxygen. I'll talk about the rest of them. Again, intravesicular microbes and antigen presentation. Another things that these phagocytic cells do is presentation of antigens. So again, phagocytosis, they are phagosomes, and again, merging of lysosome with phagosome and making phagolysosome, and then cutting of the protein or antigens of the microbe to small peptides, and binding of that peptide to the MHC class 2, and presentation of antigens to CD4 T cells. This is another way that has some consequences that you will saw and you know. So that was the phagocytic cell. They are microbes, phagolysosomes, cutting of the microbe to small pieces, some of them on the MSC class 2, and this one is CD4 positive cell. So we know that here we have co-stimulators. And as a result of binding of co-stimulator to the ligand, we will have activation of CD4 T cell and clonal expansion. Okay? Proliferation. As a result of interleukin 2 production. I told you that interleukin 2 is very important for clonal expansion of lymphocytes. Then we will have interleukin 12 when the antigen is intravesicular. When the antigen is intravesicular, the phagocytic cell will produce interleukin 12. And interleukin 12, have a look here. Let's go again step by step. It's important. It's the summary of many things that we have talked about until now. So the first step, have a look here. We have a phagocytic cell, for example macrophage, that we know that macrophage is an APC. So the macrophage is phagocytosing the microbe. So the microbe gets phagocytosed here. After phagocytosis, we have merging of lysosomes with phagosome. These vesicles are called phagosome. Now we have merging of lysosome with them, which is called now phagolysosome. And the bacteria is being killed, and the lysosome enzymes cut the proteins of the bacteria to a small piece or peptides. And then we will have expression of that peptides on the cleft of MHC class 2. Is that clear?
Yeah.
For CD4 T cells. And this APC will express co-stimulator B7 CD28. Then we will have activation, production of interleukin 2, and when we have interleukin 2 will induce proliferation, clonal expansion. And again, over time, macrophage will produce interleukin 12. Do you remember interleukin 12 as type 1 cytokines? Interleukin 12 will induce differentiation of this T helper to T helper 1. Because the antigen is intracellular, so we need T helper 1. So what determines here the type of T helper? Interleukin 12. So interleukin 12 is an important inducer of T helper 1. Okay? Interleukin 12 is an important inducer of T helper 1. As a response to that interleukin 12 and differentiation to T helper 1, then T helper 1 will produce interferon gamma. Interferon gamma, I told you, that is a macrophage activating cytokine. It will increase the activity of macrophage. So, and besides, what we have here? No. That fast and fast again is for regulatory T cell and cytotoxic T cell. CD40 and CD40 ligand. So, you know, this one provides signal for T helper, and this one provides signal for T helper, and those cytokines for, we have 3 signals for T helper 1. Interleukin 12, TCR, and co-stimulators. But these two, CD40 and CD40 ligand, will provide signal for macrophage besides these 2 cytokines. So there is a crosstalk between those cells. There is a talking between those cells. They are sending messages to each other. I've told you, cytokines are indeed messengers of the cells.
CD40 and CD40 take message to the macrophage?
Yes. Indeed, T helper 1, by production of interferon gamma and expression of CD40 ligand, wants to help the macrophage. APC is activating T helper 1, and T helper 1 is going to activate macrophage, increase the activity of macrophage. The next step, so we have enhanced microbicidal activity. Microbicidal means
Killing
killing properties, killing of the bacteria. What does that mean? Production of oxygen dependent. The previous one was oxygen independent killing mechanisms of APC or microbes. Here we have some oxygen products, superoxide or other products of antigen that they are called reactive oxygen species. You've heard about it. Reactive oxygen species. They are harmful for bacteria. They can kill the bacteria. Before T cell help, the macrophage cannot produce a significant amount of those materials that they are toxic to the bacteria. But after receiving help by CD40 ligand and cytokine interferon gamma, macrophage can produce reactive oxygen species. Okay? Clear or not? This is called oxygen dependent killing. Killing by the content of lysosome was called oxygen independent killing. Until now, we have talked about two mechanisms. Oxygen independent killing that it doesn't need for T cell, T helper 1 help. But another mechanism is oxygen dependent killing that it needs T helper 1 helps or signals. And on the other hand, we have NK cells, another important cell in intracellular pathogens. Do you remember? So NK cells will again, this interleukin 12 can increase the activity of NK cell, and NK cell will produce interferon gamma. And again, we will have another level of enhancement of macrophage activity. Clear or not? So the first mechanism was phagolysosome formation. We are talking about innate immune response and of course adaptive immune response against intravesicular. We are talking just about intravesicular pathogens. So the first mechanism was phagolysosome formation. The second was T helper mediated macrophage activation or enhancement in macrophage activation. And the third mechanism was NK cell mediated macrophage activation. Those are three mechanisms by which, with the cooperation of innate and adaptive cells against intravesicular microbes. No question? So, let me check my slides. Have a break. Please, just 10 minutes. Please.